I'd like to introduce the next speaker. It's Dr. Manoji-san. It's from the USM. He's also talking about the bioplastic from the palm oil trunks using the SAP. Please, if so, Manoji-san, so ready to your presentation, please. To your presentation.
Good evening. Yeah, I mean, I've sent my slide recording to Ota-san because I can't share my screen here.
So
you can share that, Manoj. You can share.
I can share. All right.
Yeah, you can share. Midani, just allow him to share.
Okay, sure.
Stop this sharing and allow him to share the screen.
Okay, so
yes, I mean he can share now.
Yeah, Manoj. Yeah.
So I've shared mine. So
yes.
Can you see my presentation?
Yes, because if you are present, we like you to direct, to communicate instead of recording. We face a lot of issue.
Okay.
Yeah, it's good. The visible that your slide is visible to us.
All right. Okay, so I'll start my presentation.
Yes, Manoj, go ahead.
Yeah, please. So we cannot hear you. Manoj, we cannot.
Manoj, we don't hear you.
Oh, you can hear me?
Yes, yeah. Now yeah.
No.
Wait, I think I'll mute my mic.
Yeah, please. So
no, you mute yourself, unmute. Yeah, now it's okay. Again gone your voice. Just now we listen your voice,
You can still hear me?
Yes, yes, very clear now, very clear.
The slide recording you can hear, right?
Yeah.
All right, okay.
You just go to your slide.
Okay, so let me just interrupt for a second. So you're playing a recorded presentation?
Yes, I am playing a recorded presentation.
Yes, I didn't notice that. I thought that you will present yourself. So I may ask you to stop the screen sharing.
You stop the Manoj your screen now.
Yes, and when you start again, you start again the screen sharing, please click before you select the screen. Please click on share sound and optimize for video clip. Okay, and also please use the computer speaker, not the headphones.
Okay. Okay, so before I share, I have to
Midani, can you again give the instruction to him?
Yes, so you just click on share audio and optimize for video clip.
Okay, I get it. All right.
And play the voice from the speaker and don't mute yourself.
Okay, understood. Okay, so I'm sharing now. So you can hear. Before we begin with today's presentation, let me give you a brief introduction to oil palm trunk, or also known as OPT. In general, the economic lifespan of oil palm trees are between 20 to 25 years, after which the productivity of the oil palm trees starts to reduce. In order to offset this, the old oil palm trees are locked and chipped into smaller pieces. It is estimated at least 100 tons per hectare of OPT is generated from plantations in every replanting cycle. So that is a huge biomass if it is accumulated throughout the country. What is done with this huge biomass? In a common practice, the OPTs are left in the plantation in windrows or scattered on soil for nutrient recycling. So what you see here, this picture is an example of windrow. The OPTs are cut into smaller pieces, chipped and arranged in rows between the trees for nutrient recycling. The OPTs are considered unsuitable for timber wood. This is because due to the high water content in the inner trunk region, which makes it a softwood and cannot be used for any furniture production or for timber wood in general. But with constant research and development practices, the OPT fibers are being used for the production of particle board, composite, and also made as pellet fuels. As I've mentioned previously, OPTs are considered unsuitable for timberwood production due to the high moisture content in the inner middle region of the trunk. It is reported that the inner middle region of OPT consists of more than 80% moisture. When the OPTs are felled and chopped into small cubes like what you're seeing here and pressed using a hydraulic press, this is a customized hydraulic press that is meant to accommodate the OPTs. So this is the close-up of the hydraulic press. So the OPT cubes are placed in this chamber and pressed using a high pressure hydraulics. So when these OPT cubes are pressed, the liquid, the 80% liquid that is trapped in the inner middle region of the trunk is released, and it gives us a yellow liquid, also known as OPT sap. It is reported that the OPT sap consists of major sugars such as glucose, sucrose, fructose, minerals, organic acids, and amino acids. But among all the major sugars, glucose was found to be the most dominant sugar that is present in the OPT sap. So this makes the OPT sap as a very suitable medium for fermentation. But OPT sap is an underutilized substrate for fermentation. While there are reports which has been published showing that the OPTs can be used for the production of lactic acid and bioethanol, there are less studies which have utilized OPT sap for the production of microbial bioplastics. I will talk more about microbial bioplastics in my next slide. Now I will be talking about microbial bioplastics, or specifically polyhydroxyalkanoates, or PHAs. PHAs are a type of microbial polyester which is produced by microbes as an energy storage compound. In a normal scenario, microbes thrive with ample of carbon, nitrogen, oxygen, and microelements around it. In the event when one of the elements, such as nitrogen, starts to deplete, and when there's excess of carbon around the bacterial cells, the bacterial cell starts to go into stress. So in the event of stress, the bacterial cell starts to utilize the excess carbon that is available around it and converts them into polymer inclusions known as PHA. So this PHA is accumulated inside the bacterial cells and is slowly utilized by bacteria for its survival. The conversion of these carbon sources into PHA granules is conducted by PHA synthase gene, which is present in some bacterial species, some microalgae, and archaea. The common carbon sources that are used for the production of PHA is oil, so it can be plant or animal oils, or sugars. So the PHAs that are accumulated inside the bacterial cells, when it's extracted out by breaking down the bacterial cells, looks like a white spongy material. So this raw extracted PHA it can be fabricated into various forms and for various applications. So what you're seeing here is the raw extracted PHA has been fabricated into plastic-like films of various sizes and various thicknesses in our laboratory. The main characteristic of PHA is it is completely biodegradable. So if you place a PHA film on a soil surface, it gets completely biodegraded within a span of 3 to 4 months and goes off, and there are no traces in the soil after a span of 6 months. And PHA is thermoplastic, where it has high processing temperature, and is also biocompatible, where it can be used for various scaffolds and implantable devices in various biomedical applications. The physicochemical characteristics of PHA resembles to some petroleum-based plastics such as polypropylene, making it a good alternative for petroleum-based plastics. So OPT sap, which has a high content of sugar, it can be utilized as a potential carbon substrate for the production of PHA. So that brings us to the next step. So the next step is for us to investigate, is OPT sap a suitable medium for the production of PHA. This brings us to the experimental approach. The main aim of this study is to evaluate the use of OPT sap as the carbon source or cultivation medium for PHA production using Cupriavidus necator NSDG mutant. The main reason why we are using Cupriavidus necator as the choice of bacterial strain for this study is because this strain is the model PHA producing strain, and it is also the best PHA producing strain that is widely studied around the world. The downside is Cupriavidus necator wild type strain is not able to utilize glucose, which is the dominant sugar that is present in OPT sap, for the production of PHA. Therefore, we opted to use the Cupriavidus necator NSDG mutant, which is able to utilize glucose instead of fructose as the sole carbon source for the production of PHA. This strain was developed by Professor Fukui's team in Tokyo Institute of Technology, and we are very thankful to him to receive this strain for this experiment. This study is divided into 3 stages, and we have devised 3 simple experiments to study the effects of OPT at various levels. The first stage is to study the effect of various sterilization methods of OPT sap on PHA biosynthesis. When the OPT sap is extracted, it is done in an open environment, and definitely it introduces a lot of microbes into the OPT sap. Therefore, the OPT sap has to be sterilized prior for any usage in PHA biosynthesis experiment. Therefore, we identified 3 different sterilization methods. The first one is filter sterilization. The second one is modified steam sterilization, which uses autoclave, but at lower temperature, so, which is 110 degrees for 15 minutes. And the third one is the standard steam sterilization, so which is done at 121 degrees for 20 minutes. So once the conditions here, the best condition is selected, then we move on to the second stage, which is the studies on the utilization of OPT sap as a sole carbon source for PHA biosynthesis. So here we are in the first experiment, we are studying various OPT sap concentrations based on total sugar content. So we have a range of total sugar concentrations that was set from 5 to 25 gram per liter. So once this condition, the best condition is selected, then we study the effects of changing the nitrogen source concentrations at various concentrations as well, from 0.34 to 0.74 gram per liters as well. And we wanted to see the effects on the PHA production as well. So after this stage, the third stage is to study on the utilization of OPT sap as a cultivation medium for PHA biosynthesis. In the previous stage, we used OPT as the sole carbon source. In this stage, in this stage 2, the bacteria was cultivated in a PHA biosynthesis medium. But in the third stage, the OPT itself was used as a cultivation medium for PHA biosynthesis. So this was done as a replacement to the standard mineral medium for PHA biosynthesis with OPT sap. And the total sugar content in the final medium was varied between 5 to 25 gram per liters. Now we will look at the results. So here you are seeing the results from the first stage of experiment, so which is the study on the effect of OPT sap sterilization methods. As I mentioned, we selected 3 different sterilization methods. The first one is filter sterilization, modified steam sterilization, and standard steam sterilization. For filter sterilization, the OPT sap was passed through standard syringe filter with 0.22 micron pore size. For modified steam sterilization was autoclaved at a lower temperature compared and a shorter time compared to standard sterilization method, which is 121 degrees for 20 minutes. The PHA content that was produced from this experiment is relatively low because we did not optimize the total sugar concentration in the study. So we started off with 10 gram per liter for this study. So that is the reason why the PHA content is not so high in this study. But let's look at the cell dry weight. So the cell dry weight sees some marked differences between each sterilization method. So particularly the standard steam sterilization method gave the lowest cell dry weight compared to filter sterilization and modified steam sterilization, which almost falls in the same range between 5 to 6 gram per liters. So the reason could have been because when the OPT sap was autoclaved at a high temperature, the sugars that are present inside the OPT sap may have been caramelized, causing the sugars are unavailable for the bacteria to be utilized. So this may have given the low cell dry weight in the bacterial culture, and also this bacterial strain that is studied produces 3-hydroxybutyrate as the type of PHA. But of course, there are many other types of PHA that can be produced by changing the type of carbon source and the supplements that are given to the bacterial strain. But for this study, we have stick to only 3-hydroxybutyrate. A comparison was also made between the growth and sugar consumption of the bacterial strain used in the study for both the filter sterilized and the modified steam sterilized saps. We did not include the standard steam sterilized sap for these studies because of the low cell dry weight that it produced. So we have opted that out, and we have compared the modified steam sterilization and filter sterilized sap. As you can see, the sugar consumption of the bacterial strain along with the growth showed no significant differences between each other. So this has also confirmed that either steam sterilization, the modified steam sterilization, or the filter sterilization can be used for our subsequent steps, for our subsequent experiments. But we chose modified steam sterilization as the method of choice because it is easier to sterilize the sap using autoclave compared to filter sterilization, which is much more difficult to be conducted. We are now in the next stage of experiment, which is the biosynthesis of PHA using various concentrations of OPT sap as the sole carbon source. For this experiment, we have set a range of total sugar concentration in sap between 5 to 25 gram per liter, and this sap was added to a standard PHA biosynthesis medium, which is the mineral medium, as the sole carbon source, and incubated for 48 hours at standard incubation temperature and shaking speed before it was harvested and subjected for further testing. As you can see here, the sugar concentration of 20 gram per liter showed the highest cell dry weight, which is about 8.3 gram per liter, and also high PHA content, which is about 26% compared to all the other concentrations, total sugar concentrations that were tested. And of course, this is not yet the optimized condition. So once we get the best condition here, the 20 gram per liter, so we have subjected this condition to the next stage of experiment, the next step, which is to vary the nitrogen concentration and to see the effects in the PHA accumulations. Okay, now we come to the next step of the experiment, which is biosynthesis of PHA using various concentrations of nitrogen source and OPT sap as the sole carbon source. In this experiment, we have maintained the total sugar concentration of OPT sap at 20 gram per liter, while changing the concentration of nitrogen between 0.34 to 0.74 gram per liters. So nitrogen limitation is one of the important factors in PHA biosynthesis, because nitrogen limitation is what triggers the PHA accumulation in bacteria in this case. So a good carbon to nitrogen ratio is important in order to achieve a maximum PHA accumulation profile in the bacterial cells. So coming back to the results, out of the 5 concentrations that were studied, the 0.64 gram per liter showed the highest accumulation of PHA in the bacterial cells compared to the rest of the concentrations. So this really shows that by changing the concentration of nitrogen, it really helps to increase the not just the cell dry weight, but also the PHA content, the PHA accumulation in the bacterial cells. Of course, more optimizations have to be done because this is just an initial study. More optimizations have to be done in order to achieve the best condition, the best carbon to nitrogen ratio with the best PHA accumulation profile. Having completed the second stage of experiment, which is the biosynthesis of PHA using OPT sap as the sole carbon source, we have now moved on to the third and the last stage of experiment, which is the biosynthesis of PHA using OPT sap as the main cultivation medium. So the difference between this experiment and the previous one is the previous experiment uses OPT sap as a supplement of carbon source to the standard PHA producing medium, which is the mineral medium. But in this experiment, we use OPT sap itself as the PHA biosynthesis medium, while adjusting the concentration of sugars between 5 to 25 grams per liter. So in general, what we can see is the utilization of OPT sap as the main cultivation medium shows much lower cell dry weight and PHA content accumulation compared to the utilization of PHA as the sole carbon source. Why? Because the conditions are not yet optimized. So this is just the start, but rest assured, we can see that the OPT sap can be used as the main cultivation medium. So judging from the various total sugar concentrations that we use, so we found that 20 gram per liter of OPT sap, 20 gram per liter of total sugar concentration could give us a decent amount of PHA content, which is about 22% with 5.8 gram per liter of cell dry weight. But of course, a lot more optimizations has to be done in order to optimize the conditions, the compositions of the medium to maximize the production of PHA using only OPT sap as the main medium. This brings us to the conclusion of the presentation today. So from this experiment, we found that OPT sap was successfully utilized by the bacterial strain Cupriavidus necator NSDG mutant to produce PHA. So the mutant strain was able to utilize the dominant sugar inside the OPT sap, which is glucose, and convert them to PHA. Besides that, the OPT sap can be used both as sole carbon source and cultivation medium for PHA production. Of course, this is just a statement based on the results that we got, where it can produce PHA. But more optimizations are needed in order to maximize the conversion of sugars in OPT sap into high bacterial density and pHA accumulation. So more studies are needed in order to convert the maximum amount of sugars from OPT sap to high pHA production. So these are some of the acknowledgements I would like to make to all SATRAP's OPT research project members, Ministry of Higher Education, JIRCAS and JICA, Japan, School of Industrial Technology, USM, Prof. Fukui from Tokyo Institute of Technology, and Ms. Husna Liana for conducting the experiments in this study. Thank you very much, and I'm open for any questions.